The story begins with mRNA transcript entering the grasp of the ribosome. Out comes a primary sequence of amino acids that fold into tertiary conformation.
The tertiary conformation yields a functional protein. Wow, look at this functional protein.
Ribosomes translate mRNA in the cytosol and on the surface of the rough endoplasmic reticulum to make a protein product. Part of the protein contains a signal sequence that is identified by an SRP, or signal recognition particle, that binds to the protein and the ribosome. The SRP receptor on the surface of the endoplasmic reticulum recognizes the SRP and allows the protein to enter the ER. There are two types of proteins that can be let into the ER. There are water-soluble proteins that go all the way through the ER membrane and are released into the ER lumen. There are also proteins that do not pass all the way through the membrane, but instead are partially anchored in the ER membrane.
Here, a transporter protein will allow our functional protein product to enter the endoplasmic reticulum. Chaperone proteins assist in the folding process by identifying misfolded proteins and blocking their passage out of the endoplasmic reticulum. They also will permit functional, properly folded proteins to exit the endoplasmic reticulum and enter the rest of the cell.
The signal sequence is always taken out of the protein when it has been sorted and the protein modification, including the attachment of an oligosaccharide chain, occurs in the ER. Chaperonins pull proteins across the membrane. Misfolded proteins bind to chaperonins in the ER until the protein is properly folded. Protein enters into the COP-coated vesicles and has a new signal sequence attached to it so it can enter the Golgi. Once the vesicle enters the Golgi, it docks to the membrane using a snare, which docks the vesicle in place. The snare binds to its complement on the membrane of the Golgi, and the vesicle opens up, releasing its contents into the Golgi.
Here, we watch the journey of our functional protein product as it enters the Golgi apparatus for further processing. The Golgi acts as the sorting and processing center of the cell, directing proteins to their final destinations, which may be lysosomes, the plasma membrane, or the cell exterior if the protein is not being retained within the cytosol.
Vesicles bud from the trans-Golgi and fuse with the plasma membrane in exocytosis, which creates new lipids and proteins. The protein can be anchored in the plasma membrane or be released outside of the membrane into the ECM.
Vesicles containing functional protein products destined for the extracellular environment may travel a long way to their destinations. They will be supported by microtubules that run along the cytoskeletal filaments within the cell. Here, we watch our functional protein product being transported to the outside of the cell. As the cell nears the plasma membrane, proteins will be recruited to assist with its binding and eventual release of our protein product.
Endocytic vesicles trap molecules that are in the extracellular fluid and bring them into the cell. Macromolecules in the extracellular fluid bind to receptors on the cell surface and enter as complexes in vesicles. The vesicles carrying the protein fuses with the endosome. The endosome acts as a sorting station for the endocytic pathway. At this point, there are three places for the protein to be transported. It can be sent back to the plasma membrane, it can be sent to the lysosome, or it can be sent to a different domain in the plasma membrane.
Much to our protein's dismay, their time of function is up. Thus, they will be transported into the lysosome, where their destruction into basic amino acid components will be undergone in a strongly acidic environment.
Lysosomes are where the protein finally get degraded through hydrolytic enzymes. The lysosome contains an ATP-driven H+ pump, which utilizes ATP to move H+ into the lysosome.